Hello friends, welcome to B.Sc.Agriculture. If you are coming to my channel for the first time, then don't forget to hit the subscribe button. Your subscription and likes are always motivating us. If you have any doubt, please comment in the section. So let's move to the video. In this video we are going to see about fundamentals of pathology. I am going to cover the general characteristics of fungi as well as the mycelial character as well as the vegetative resting structures, which is said to be known as the spores. So while seeing about the fungi, so which used to have Thallus, mycelium. So first I am going to describe about the Thallus of fungi. So which is said to be known as the vegetative body of the fungi. It used to consist of elongated continuous tubular or hollow filamentous structures. So we can able to call the individual filament called as hyphae. So after that we are going to see about mycelium. So the collection of hyphal bodies are said to be known as the mycelium. So based on the presence of the separating lines, they can be called as aseptate mycelium as well as septate mycelium. So the aseptate is where you can able to see the absence of lines, and in septate you can able to have the presence of lines. And while seeing about types of thallus, so the first one is holocorpic, so which is said to be the non-mycelial type. So the entire thallus is said to be converted into one or more reproductive units. So while in comparison, the somatic as well as the reproductive unit does not occur, which is as same as in this individual. So the example is Syncytium endiobiotum, which is also known as yeast. And after that we are going to see is eukarpic, so which is a mycelial type. So the portion of thallus is converted into reproductive structures, for example Pythium and Phytophthora and Plasmodium, so which is said to be known as the naked motile as well as multinucleate mass of protoplast, which used to feeds on an amoeboid fashion. So example Plasmodiphora basicae. So while seeing about the color of fungi, they are said to be usually white in color, so they are said to be called as hyaline in nature, and occasionally they are said to be brown or black, and it's due to the shade of pigments of the color. So you can able to see the color of the fungi as two types, the hyaline as well as the colored ones. And while seeing about the types of mycelium, they are ectophytic, endophytic, ectoendophytic, intracellular as well as intercellular. So the ectophytic is said to be the one of the mycelium which is used to present on the outermost surface of the host, example Irregular, and in endophytic, which is used to penetrate the host and develop into the tissue, so which is said to be known as the coltotrichum and rhizopus. So the ecto and endophytic, so which are said to be present on both surface as well as within the host wall. And while seeing about the intracellular as well as the intracellular, so the intracellular is the one which is where the mycelium penetrates in between the cells, as well as the intracellular is said to be known as the where the mycelium used to grows inside the cell. And while seeing about the types of mycelium, they are said to be monokaryotic mycelium or haplomycelium. So the single nucleus cell present or dikaryotic mycelium. So the mycelium with the two sexual different compatible nuclei per cells. And while seeing about the homokaryotic mycelium, they are said to be genetically identical, present with the genetically identical nuclei. So and the heterokaryotic mycelium is where the nuclei used to have different genetical mediums. And whilst we are going to see about the types of mycelia, as well as I have already defined those, but I am going to give you the pictures. So the intracellular as well as intercellular, so they are said to be present inside the cell, which is intracellular, as well as present in between the cell, which are intercellular. So after that we are going to see the special stomatic structures of the fungi. So the first one is aprosorium, so which is said to be known as the simple bilobed swelling of the germ tube, which is used to attach on the surface of the host cell. And the hastoria, which is different from the aprosorium in slight manner, which is used to penetrate the cell to absorb food material from the host, so which is used to act as a root for the microorganisms. And the rhizoids, so which is said to be a short root-like filamentous outgrowth without the nuclei, which is used for anchoring as well as absorbing organ, so which is as same as or else as like as Hostoria. So after that we are going to see the different modifications of fungal hyphae. So Pletencyma, or you can able to name it as fungal tissue, where the mycelium of higher fungi organized into woven tissues, so which is said to be distinct from the loose hyphae of Thallus. So it's how you can able to define a fungal tissue. And while seeing about the different types, or else you can able to call it as the modification. The first one we are going to see is Prosenchyma. They are said to be the loosely woven fungal tissues which used to have individual like parallel to one another, and they can ably said to be easily distinguishable. And after that we are going to see is Pseudoparenchyma, where the compactly woven fungal tissue with the isometric or oval cells resembling parenchyma are said to be indistinguishable in the Pseudoparenchymas. And after that we are going to see the modification of fungal hyphae. So the first one is Rhizopus, which used to seem like a long root-like standard of closely packed parallel hyphae forming compact mosses. So example you can call Ganoderma. And after that we are going to see the modifications of fungal hyphae continuation. So where you are going to see the stroma. So it is said to be a compact vegetative structure of fungi which is said to be often bearing sporocarpus, either which is said to be present upon the spores of the stroma. Example Claviceps, Fusarium and Cultotrichum. And after that we are going to see Sclerotia. And while seeing about the reproductions of fungi, there are said to be three types. So the vegetative one, asexual, as well as the sexual one. So as the name indicates, the vegetative, which is the modification of hyphal cells or mycelium, and they can able to produce the new one. And the asexual, where there is no union of male or female gametes, so they can able to go for the reproductive functions like binary fission. And in sexual, where the union of male and female gametes are said to be takes place and the formation of younger ones. And while seeing, we are going to first see about the vegetative spores. So the first one is Sclerotia, where the compact hard mass of dormant mycelium with varying shapes and size are said to be formed. So they are said to be brown, black, or out on outer side, as well as the grey or white on the inner sides. So they're said to be mainly resistant for unfavorable conditions, as well as dormant for long period of time until there's suitable climate occurs, so they can able to sprout and emerge on the host surface. So while seeing about the Sclerotia, they are even classified as two types, which is regular one as well as irregular one. So in regular one there are said to be two types, so we can able to classify them based on their color, so which is the brown as well as black. So I have given examples for based on the color. I have given examples like Sclerotium rolfsi, Sclerotium oryzae. So some of the example organisms. So after that there is said to be a regular brown, which is Sclerotium rolfsi, as well as Claviceps microcephalus. You can able to see the pictures of them. And after that, so the regular black, which is Sclerotium oryzae, and irregular brown, which is Rhizoctonia solani, as well as irregular black, which is Rhizoctonia bacticola, as well as Sclerotia and Sclerotiarum. So after that we are going to see about the types of Sclerotia, so which is Mycelia-genus. So the first one, based on the name indicates, they used to form a thread-like spindles present on their appearance. And after that we are going to see Carpogenous claviceps. So they used to have a spore of mushroom-like structures on their one, and sporangious, so they used to form a fungal hyphae tree-like structures based on that. And after that we are going to see about the Chasmode Chlamydospores. So a Chlamydospores is said to be a thick-walled and resistant structure of fungi which are said to be produced even under the unfavorable conditions of the host as well as the parent organisms. They are said to be usually dark colored and spherical shaped in them, and they are terminal as well as the intercalary in nature. Example Fusarium. So these are the pictures of Chlamydospores where you can able to see the terminal as well as the intercalary spores. And after that we are going to see about the methods of asexual production, which is the fragmentation is the first one. So it in this process where the mycelium used to breaks into two or more similar fragments, which is mainly done due to the external force. So after that application of external force, each fragment is said to be grown into new mycelium forms. And after that we are going to see about the fission. So the parent cells, which used to be split into two equal halves, each of which used to develop into new individuals. Example, yeast. And fragmentation. So in fragmentation, it's like we commonly see in the plants. So the vegetative cells usually break through the aerial hyphae, as well as the functional reproductive bodies are said to be spread by air. And you must need to know one important character, where these functional reproductive bodies, which it does not use to have a resting period, so they must need to find the suitable host within a particular time, so they can able to reproduce. So we can able to call them as arthrospores. Example, powdery mildew. And after that the third one is budding. So where a smaller part of the cell is said to be used migrated from the parent cells, so which is almost used to contain the characteristics of the mother cell, which used to detach and grow into a new individual. So the budding is said to be most common in unicellular forms like yeast. And while seeing about the production of asexual spores, the first one we are going to see is endogenous. So the spores produced inside the sacs are said to be in sac-like structure. So this sac is said to be called as sporangium. The spores may be motile as well as the immotile one. So the motile spores are said to be known as the zoospores, and immotile spores, which are said to be known as the aplonospores. So the zoospores example is Phytophthora as well as Pythium, and then aplonospores we can able to, the example organisms are Mucor as well as Rhizopus. And in Exogenous, they used to have conidia structure, and they are said to be produced through a specialized structure, which is also nakedly structured, is said to be known as the conidiophore. They can be either a single as well as this chain of conidia are said to be formed in the conidiophore, and they use to have varying form and size based on their locality, and they can be either unicellular or multicellular, as well as colored or hyaline, as well as but they are said to be non-motile in nature. So these are the pictures of conidia. So you can able to see the pictures of conidiophore as well as the conidia, spores of conidia, which is said to be formed in the conidiophores as well as spreading. So after that we are going to see about the fungal diseases, which are we can able, I have listed out some of them, and we can able to see one by them in future classes. So I have given the lecture notes in description. So if you have any doubt, please comment in the section, as well as join us in the Instagram page. So if you like the video, like it and share it with your friends, and subscribe to B.Sc.Agriculture and hit the bell icon to get regular updates. So thanks for watching the video. I'll catch you up in next videos.